Welcome to this lecture on fundamentals of genetics. Today, we are going to explore a fascinating topic called extra-chromosomal inheritance. Most of the time, when we think about DNA and genetics, we picture chromosomes neatly packed inside the nucleus of a cell. But what if I told you that some of your most crucial genetic instructions are actually floating outside the nucleus? Understanding this hidden genetics is essential for everything from tracing human evolution to diagnosing rare diseases. Let us dive in. To understand how this works, imagine you are building a highly complex robot. The main instruction manual is kept safely in a central control room. This central manual is just like the DNA inside the nucleus of a cell, dictating almost everything about how the robot is built. However, imagine that the battery packs and solar panels came with their own tiny separate instruction booklets taped directly to them. In biology, this is exactly what happens with energy-producing organelles like mitochondria and chloroplasts. They have their own separate DNA, completely independent of the main nuclear DNA. Now that we have our analogy, let us look at the biological definition. Extra-chromosomal inheritance, also known as cytoplasmic inheritance, refers to the transmission of genetic information that is not located within the nucleus. In standard Mendelian genetics, both parents contribute equally to the offspring's chromosomes. But here, the rules change. Because the mother's egg cell is typically much larger than the sperm cell, it provides almost all the cytoplasm for the new zygote. As a result, these organelle genes are inherited almost exclusively from the mother, creating a very strong maternal lineage bias. This unique form of inheritance is not just a biological quirk, it has profound real-world implications. Understanding extra-chromosomal inheritance is absolutely critical because mutations in these non-nuclear genes can cause severe, often fatal diseases. For instance, mitochondrial diseases primarily affect organs that require massive amounts of energy, like the brain and muscles, leading to conditions such as sudden blindness. Beyond human health, plant breeders use this concept to easily produce hybrid crops. Furthermore, because mitochondrial DNA is passed down almost unchanged from mother to child, scientists use it as a genetic time machine to trace human evolutionary history back thousands of years. Since we know these diseases are passed through the mother's cytoplasm, we need a way to measure their impact. This brings us to the concept of heteroplasmy. The formula you see here calculates the severity of an extranuclear disease. S represents the severity of the physical symptoms expressed by the offspring, measured as a percentage. NM is the number of mutant or defective organelles inherited, while NT is the total number of those organelles present in the cell. Conceptually, this tells us that disease severity is directly proportional to the fraction of defective organelles. If the number of mutant organelles increases while the total stays the same, the severity goes up, and the offspring will show stronger symptoms. Let us apply this formula to a concrete example. We are given a mother who carries a mitochondrial mutation for muscle weakness. She has a total of 5,000 mitochondria, and 3,500 of them are mutated. The father has normal mitochondria, but we completely ignore his contribution because this is extra-chromosomal inheritance. The threshold for showing symptoms is 60%. We substitute our values into the heteroplasmy formula, dividing 3,500 by 5,000, which gives us 0.70. Multiplying by 100 gives a final severity of 70%. Because 70 is strictly greater than the 60% threshold, we can conclude that the child will indeed suffer from muscle weakness. Having seen how mitochondrial ratios affect disease, let us look at another fascinating phenomenon called maternal effect. A classic example is the shell coiling direction in the snail Limnia peregra. Here, a dominant allele D causes right-handed coiling, and a recessive allele, little d, causes left-handed coiling. But there is a twist. In standard genetics, an offspring's own genes determine its physical traits. In maternal effect, however, the phenotype of the offspring is determined strictly and exclusively by the genotype of the mother. The offspring's own genes and the father's genes have absolutely zero influence on this specific trait. To formalize this concept, we use a very straightforward logical formula. PO equals F of GM. In plain English, this states that the physical appearance, or phenotype, of the offspring, represented by PO, is a direct function of the mother's genetic makeup, represented by GM. Notice what is missing from this equation. The variables for the father's genotype and the offspring's own genotype are completely absent. This is a radical departure from standard genetics. It forces us to look exclusively at the maternal generation to predict the physical traits of the current generation. Let us put this maternal effect formula to the test with an example. Imagine a female snail that physically has a left-handed shell, but her hidden genotype is heterozygous, meaning she carries one dominant D and one recessive D allele. She mates with a male who has a right-handed shell and a recessive genotype. What will the babies look like? Following our formula, we completely ignore the father. We also ignore the mother's physical shell. We only look at her genotype, which is DD. Because the D allele is dominant, it dictates the outcome. Therefore, the final phenotype value is right-handed. All of her offspring will have right-handed shells, regardless of their own genetics. Now that we have calculated the result, let us prove why this biological mechanism works. Why does the mother's genotype override everything else? It starts with the mother's heterozygous genotype. Because she possesses the dominant D allele, her cells actively manufacture right-handed determining proteins. Here is the crucial step. The mother pumps these specific proteins directly into the cytoplasm of her eggs, long before fertilization even occurs. By the time the sperm arrives, the egg is already preloaded with instructions. These maternal proteins dictate the developmental direction of the embryo from the very first cell division. This beautifully proves how the maternal environment in the egg completely overrides the offspring's own nuclear DNA during early development. To solidify everything we have discussed, let us look at this diagram comparing the two types of inheritance. On the left side, you see standard nuclear inheritance. Notice how both the sperm and the egg contribute chromosomes, colored blue and red, which fuse together in the zygote's nucleus. This is a biparental process. Now, look at the right side, showing extra-chromosomal inheritance. The sperm is small and contributes almost no cytoplasm. The egg, however, is massive and packed with green mitochondria. When they fuse, the resulting zygote inherits all of its green mitochondria exclusively from the mother's egg. This visual perfectly captures why these traits follow a strict maternal lineage. We have covered a lot of ground today, so let us summarize our key takeaways. First, extra-chromosomal inheritance breaks traditional Mendelian rules because it relies on DNA found in organelles like mitochondria, not the nucleus. Second, because the mother provides the vast majority of the cytoplasm, these traits are passed down almost exclusively through the maternal line. We also learned that the severity of these traits depends on heteroplasmy, which is the ratio of mutant to normal organelles. Finally, in cases of maternal effect, the mother's genotype alone dictates the offspring's physical appearance. Understanding these concepts is absolutely vital for diagnosing complex metabolic diseases and tracing our evolutionary history. Thank you for watching.